Hello and welcome back to Better Biopharma, the official podcast of Bioprocess Online. I'm your host, Tyler Menichiello, and today I'm joined by Dr. Sean Tucker, the founder, Senior Vice President, and Chief Scientific Officer of Vaxart, a company that is developing oral recombinant pill vaccines. Dr. Tucker, thank you so much for being here.
Thanks for having me. I really appreciate it.
No problem. It's my pleasure. So to get started here, why don't you tell our audience a little bit about Vaxart and the products that you have in development, specifically these oral recombinant vaccines?
Yeah. So the reason why we started the company is that we wanted to build a vaccine technology where the vaccine could come to you rather than having to go and get a shot, whether you have to wait in line or wait for your doctor. We thought that, if you could provide something to everyone's household in the US, and the idea was that we could send it by mail or drone or whatever sort of technology you'd want, and it comes to you. And for us, a tablet is the simplest technology you could possibly have because everybody knows how to take a tablet. So our idea was to build vaccines that we could send in tablets. And it took us a while to get there, but we have vaccines now in candidates, I should say, where for norovirus, for COVID, for flu, and we have a therapeutic vaccine for HPV. So those are the things we have in development right now.
Yeah. That's great. Thank you. I'm always fascinated when I hear about oral biologics because I think it really represents an innovative hotbed of development right now in the biopharma space. And I'm curious. I mean, you mentioned accessibility as one of the reasons for pursuing this oral formulation, but were there any other motivations behind that as opposed to taking a traditional like injectable vaccine route?
Well, I think that what's probably been underappreciated is that when you inject a vaccine, you get an antibody response in your blood. And so it doesn't necessarily make antibody responses in your nose and your mouth or your intestine. And there are definitely pathogens that invade through your nose, your mouth, and the mucosal systems. And what we found is that our vaccine tablets could elicit, your systemic response, the blood, but they also could put antibodies in the places that you're most that's basically the first line defense against pathogens that invade. And that could be for our tablet vaccine for COVID and flu. We know that we make antibodies in the nose. For our norovirus vaccine that where the norovirus invades your intestines, we basically have a way of essentially making antibodies at the place that matters in the intestinal space. And I think that's probably been underappreciated from the field because everyone tries to study the vaccines from the standpoint of I inject, I measure it in the serum. That's all I need to know. And reality is your immune system works a lot more complicated ways than just serum antibodies.
Thank you for breaking that down. And for people out there, forgive me if you already said the names of the assets you have in development, but for those at home that want to do a deeper dive and look into your products, what are the names of your assets and whereabouts are they in development?
Yeah, I don't think we've named our assets in ways that are like very creative, other than saying, here's our COVID-19 vaccine, here's our influenza seasonal vaccine. They're on phase 2 of development. Our norovirus vaccine has just finished the next generation norovirus vaccine just finished a phase 1, and we're about ready to start a phase 2 study. So I'd say that we are a phase 2 stage of development for all of our indications, other than HPV, which would be going to a phase 1 to start.
Got it. Thank you. So I do want to dive into the whole. I'm fascinated by your process development that we spoke about in our briefing call, but before then, I want to talk more about oral biologics and that approach here. How would you say Vaxart's technology differs from other orally available biologics? And I
Yeah,
think as inspiration from any other products?
well, I think that the key thing is that there's very few oral biologics. There's obviously a lot of oral small molecule that you take, Advil, whatever kind of medicines you swallow, they seem to work. But there is not a lot of oral biologics. There's a very small subset. And so for us, one of the things we had to learn how to do is essentially take this vaccine that, is normally injected, because this is how it works, and turn it into something that could actually put into tablets. So we had to work out new ingredients and new formulations to allow it to be tableted. And then we had to put enteric coatings on it so that it gets through the stomach because the biological itself is degraded by acid. And so we had to do some work to sort of get us to that point where we could actually use it and show that it works. One of the key studies is we actually had to look and see hey, what part of the small intestine or, or even the colon could be useful from the standpoint of delivering and have it open up. And we had to sort of do some work to show that it was the lower small intestine that really mattered. And so, again, all this process development work is atypical for what happens in the inject biologic space because they're mostly injected and not sort of, necessary for the standpoint of small molecules because generally you open it up anywhere, it probably does okay. So there was some work that we had to do to sort of make it go to the right place in the intestine.
Yeah. No, that makes sense. And moving into your process here. So when we spoke earlier, it sounds like there's a fair amount of overlap in the upstream part of your process, at least, to traditional biologics manufacturing. So why don't you explain a bit about that manufacturing process, specifically upstream?
Sure. So the upstream side of things, we use a non-replicating virus, but it does replicate into these cell lines that we use to allow it to grow in large quantities. We put this in a stirred tank bioreactor, which is a single-use bioreactor, so it has like a bag inside of it, which is pretty standard technology. A little propeller, it spins, it does all the things. We grow it up and then you go through a few steps of purification using ion exchange as well as EFF. And then you get to the point where you have your virus in a uh, liquid and we want to essentially dry it before we put it into tablets. And we have to go through a step of drying it. Right now, our technology uses lyophilization, which is not at standard, but people have used it for all kinds of things, including like drying out your fruit that you eat, the dried fruit that you eat. So we had to go through this drying step. And it took a little while to sort of figure out how to formulate the vaccine in a way that it doesn't allow it, so that it doesn't degrade over that time. And then, of course, once it's made in and dried, we have to figure out ways to blend it with tablet excipients. And some of these things work much better with small molecules and large biologics. We do some work and then you tablet, coat it, and it's ready to go. So there was definitely on the upstream, it's very similar to what's been done in the past for a lot of vaccines and biologics. Downstream is a little different. We had to learn as we went.
Yeah. So it sounds like the real significant change in your process is at that lyophilization step right after purifying from upstream.
That is correct. That's the part that varies from the standpoint of what's been normally done in the vaccine space with, viruses or vaccines that are virus based.
And I'm curious to hear from you what the most difficult aspects of getting that formulation right are. I'm curious, when you were developing this process, did you rely on expertise from traditional, like, small mall sector? Did you grab some pharma folks and bring them over and,
Well,
leverage their insights, or how'd that work?
yeah, it's funny because there's a lot of expertise. There's billions of tablets that are made very each year from the standpoint of small molecules, and there's not much in the way that biologics were used in tableting or capsules or anything like that. So we actually didn't really find experts that were in that space. We were able to bring in a few people that had worked on drying materials for biologics, and I think that helped from the standpoint of being able to de-risk and sort of evaluate the technologies to make it work. But yeah, we didn't bring in people who were experts in the small molecules because we thought that wouldn't necessarily be helpful.
Yeah, that makes sense. Um, and when you mentioned the drying step in other biologics, excuse me if this is a naive question, but, in what ways does that work and not work for what you guys are doing at Vaxart? Like, what were you able to pretty much take as a standard practice, and then what did you have to really build on to get that right in your process?
Yeah, I mean, I think the main thing is just on is how to operate the the lyophilizer so that you have it, you pull it under vacuum, you go to the cooling steps, and you had to spend some time trying to figure out, what the order of the operations are and the temperatures that you could operate in, and there's a couple of little tricks that we had to learn that is all trade secret, but those are the kind of things we had to go through sort of a process to go to make sure you could get it done because again, we have a product that's very atypical from the standpoint of the vaccine space, and certainly even with biologics, there's very few things that have been tableted out there.
Would you say that atypicality is part of why Vaxart decided to internalize manufacturing?
I think the key thing from the standpoint of Vaxart, we have this atypical process, and two, the other thing is that we found that there that we needed to do a lot of process development work internally to sort of get our upstream to make the sort of yields that we needed to be successful. And by not, and if you outsource it, you're going to spend a lot of time and effort having some telling somebody else, hey, try this, try this, try that. Instead, we're able to build a bigger group internally. And that was helpful to bring these people in and have them work together, come up with a plan, integrate it with the downstream. And that was super helpful from the standpoint of moving this forward.
Thank you. Thank you, Dr. Tucker. So I'm curious, was there any part of that decision being made did scale come in at all? Like, were you and or are you concerned about Vaxart's ability to scale internally? Like, once you figured out the process by, bringing in that expertise, did it come to a point where you're like, okay, well, we figured out the process, but it would just not be worth, transferring this process to a CDMO or manufacturing partner. Let's just do it in house. Like, walk me through that decision a little bit.
Yeah, I think that when we first came up with a process that was going to be useful from the standpoint of making materials for clinical trials, we realized that process was not going to be scalable. And we had to sort of change unit operations as a prop in that plan, changing out certain equipment, not doing centrifugation, in a batch way. We had to figure out ways to get around that. And I think that was very useful from the standpoint of having the people internally sort of work on this because they are and people that had expertise in doing this at large companies because they were like, yeah, you can get this done, but it's not going to be useful when you actually go to commercial. We had to basically start over on some of these things, relearn how to do things, and make it something that would be something much more scalable. And having those internal resources was very helpful from that.
Yeah, no, that makes sense. Um, I want to talk a little bit about Vaxart's history. I mean, you are the founder. What was it like trying to get funding and investor excitement around such a novel idea? Like, you have nothing to point to really on the market or in development that it's like, yeah, we're doing something like that. We're gonna make an oral vaccine. Like, I'm sure when I heard oral vaccine in a sentence, I was like, did I read that correctly? So what was that like trying to build?
Yeah, I think that again, people tend to be skeptical when because there's nothing been done before. But there was certainly a climate in the Bay Area where we started, where people were looking to innovate to try to change the way things are done that normally happen. Like everyone usually used to take taxis from the airport. Well, at some point, Uber came around and made it a lot easier to basically call up with your phone and get things going. And the same thing with when I told people about the vaccine, is like, hey, why don't we build a vaccine technology that's reusable, can be done at time and time and again with all these different indications, and have it come to your house and making it in tablets. And people were like very excited, because they thought, oh, this will change the whole dynamics of how vaccines are delivered in the future. And then, of course, the skeptics come out and they basically are saying, well, no one's been able to do it. Why is that? And so, first, you had to build your arguments. Well, we think we have got the way that's the solution that's going to work. And then, of course, eventually we were able to get the data to show that it actually worked. And so, yeah, it was a process where people were excited about the idea of doing this. And so, from that standpoint, it was easy to find people to fund it. On the other hand, you had to basically still show that you had something, that there was something there for them to invest in, that you weren't just saying, oh, I'm going to create this and have no ideas about how to get there.
Yeah, yeah. Thank you. Um, I did want to ask you another question on the process, but I'll put a pin in that right now. And along this vein of exploring kind of uncharted territory, from a regulatory perspective, what kind of boxes do you need to check, so to speak, for something like this, an oral vaccine with nothing really to compare it to? What are those conversations with regulators? What do those look like?
Yeah, I think the typical ones are they're going to ask, are there any sort of bad actors or adventitious agents that are accidentally put into your process? And so the regulators have set standards for what they expect from the standpoint of most vaccines. Where things got a little tricky is that an oral vaccine by nature doesn't need to be sterile. And so we had some conversations about, what the quality of the vaccine needs to be from the standpoint of bioburden. But we don't need to be actually sterile because you swallow things that aren't sterile all the time. And we're doing mostly okay from that. So those were some of the conversations. There have been some vaccines that have been given orally in the past, rotavirus vaccine for kids, as well as the polio vaccine was given on a sugar cube of all things, and you swallowed that. I don't know if you know that, but that's
I did not know that.
but that is the history of yeah, poliovirus was put on essentially on the sugar cube or the poliovirus vaccine and swallowed that way. So ours was a little bit different, but there was at least some indication from the, regulators that they were familiar with some oral vaccines that are out there. These are one-off, not like ours, which is a platform. And so we were able to leverage some of that to ask for different specs, like not having it to be sterile. One of the other things we were able to do is actually argue that because we're delivering things in an oral manner and you get exposed to proteins and DNA all the time, that we didn't need to have the same sort of specs on host cell DNA, host cell protein that an injected vaccine, because you're swallowing proteins and DNA all the time, and there's no ill effects. And so we felt that that was a reasonable thing to discuss with the regulators. So there's a couple things that were unique about our product compared to an injected product.
Yeah. I never, I never knew that about the polio vaccine. It's very fascinating. So I guess it's the history of Vaxart steeps pretty deep down back to, polio days. A lot, was there, and was that, and you said it was a reasonable request, and I think it's reasonable too, but was there any pushback from regulators? Like, did they try to fight you on that, or were they pretty much sold like, "Yeah, good point." Oh,
Well,
pause
I
that.
think I think the key thing about the regulators, at least the ones that we've worked with, is that they like data. So if you have data to support what you're trying to do, then they're more apt to follow along with what you're doing. And so you first have to come up with an argument why you think that, you need a different spec. And, you show data that shows there's no ill effects or that people have already been exposed to this amount of DNA. They were more apt to accept it. So and again, there's been some historical reasons that they were able to accept that as well. I believe that the poliovirus vaccine also had not as strict of specs when it came to host cell protein and DNA because again, swallowing things, intestinal exposures, you're getting proteins all the time. So
That makes sense. And you mentioned before about this being a platform, more than a one-off kind of asset. What does the future of this platform look like, and in what ways do you see it change meaningfully, like, when we talk about the process, and how does it differ from vaccine to vaccine, and in what ways do you see it taking new shapes and forms?
yeah, I think one of the key things we wanted to develop is something that wasn't just going to be a one-off vaccine that, could only work, for example, like polio. That's a replicating attenuated polio virus, and it's only useful for polio, in terms of an indication. What we wanted to build something is that could be used over and over again with any indication so that if we wanted to start and try something completely out of the blue, say Zika or tuberculosis or something, we could use the exact same technology. And the cool thing about what we're doing is that when it cassette in our antigen of choice, as we manufacture this, the doesn't seem to change whatsoever going from indication to indication. Our norovirus vaccine is made exactly the same way that our flu vaccine is made, which is made exactly the same way with our COVID vaccine. And that just makes it so much simpler as you're going forward that you don't have to spend as much time doing all this process development. It's it's indication agnostic, but it's platform specific.
Yeah. Wow, thank you. And on the topic of platforms, in an earlier episode of Better Biopharma featuring, Siren Biotechnology's Nicole Palk, she said that she thinks the pendulum is swinging from assets back to platforms in the near future. How do you feel about that? And where do you think we are right now? And, what do you think if you had to put on your forward facing predictor hat? What do you think?
I I think that people are very intrigued by new platforms, but big pharma still wants to buy what I would call a indication. They're going to want to know because they already got plenty of indications in their pipeline. They're they're looking for cool platforms, but they really want to know that your platform is going to be useful for an indication that they don't have, that they don't currently have, or can drastically improve what they've got. So I see what she's saying is that there's definitely been more interest in the platform side of things. Do you have something new and novel and interesting? But I also want to point out that, big pharma is still trying to find, indications that they can address. And if you have data that's pretty far along, it's probably an easier discussion from the standpoint of partner.
I did want to follow back up on the process. What were the most meaningful lessons you learned in not only developing the process, but scaling it to a point that's viable for what your purposes are now? And then in the future, what do you think that scaling looks like? Or are you there? Are you at the scale that will carry you through?
Yeah, I mean, right now we're operating for the most part at 50 liters. We've operated up to 250 liters. We do think that the process that we have now is scalable from the standpoint of being able to go to 1,000, 2,000 liters using the similar technology is not going to be a big change for us. I think, one of the things that I appreciate now after doing this for several years now, more than 15 on this, is that you can't shortchange your time for process development. You got to spend some time and effort to make sure that, your process is robust because while in theory, growing up, a virus in a bioreactor has been done and it's been done several times, you still have to go through a process to try to figure out what are the important parameters to sort of optimize your yields to get to the point where that it's commercially viable. Because when we started, we just wanted to show, hey, this platform works in humans and it works. But the next step immediately was to spend more time trying to figure out how to make the process work in a way that would be economically viable. And that is something that now that I've done this for a long time, I think I appreciate the fact that it just takes longer than you'd expect, even though there was definitely plenty of what I would call good actors out there that had showed the way on how to do this correctly.
And without giving away any secret sauce here, when you talk about improving yields in that early step of the process, where did you find success in that endeavor? Like, what do you think is an underlooked part of improving uh, viral replication?
Well,
And
I'll give you an example. As I think that one of the things that we had to figure out is that what cell density made the most sense, and what virus to cell ratio made the most sense to actually put in. And then from that, the timing of when you do actually do your harvest. Is it going to be 3 days later, 5 days later, 1 day later? You had to try to figure out what's the optimum time before you essentially say, we're going to stop and we're going to go and, you know, do the harvest. And that timing was really important. The ratio of the virus, this concentration of the virus, all those things really mattered a lot because there were some operating windows that were a little bit, let's just say, tighter than not. And if you didn't get to the right density, then you were going to miss your miss out on a lot big yield. And I think that's all the things that's one of the big things that I think we had to work out and work out in a way that's individual to every indication or every virus that you're working with. You can't use just, here's what I think is going to be best.
there's no one size fits all approach, I'm sure, when it comes to
No, every virus is different.
yeah. Before we move into our hallmark question, I don't want to keep you too long here, but before I do, is there any other part of Vaxart's story or process that you want to share with our audience that maybe they could learn from, or any insights you'd like to share?
I I think the key thing is just you have to spend some time, sort of working things out, and you got to be realistic that it just takes longer than you expect. And again, from our standpoint, we knew that people had grown up adenovirus in large scale. We knew that this was doable. We just had to spend some time being able to do it ourselves, and then get to the point where we felt like it was something that was going to be commercially viable. And I think that was something, that now that I've done this for several years, I think I appreciate, just how long it takes rather than, oh, this is easy. Somebody's done it already.
Mhm. Yeah, that I should have asked this earlier on. Is it a adenovirus or, associated
Yeah,
virus that you guys?
it's an adenovirus that we're using, non-replicating, at least in this target cells, but it does replicate, of course, in the cell lines we're using as we manufacture.
Mhm. I hope the audience forgives me for not asking that earlier earlier in our
Oh, no
discussion.
worries.
Well, Dr. Tuck, I really appreciate your time today, and I wanna ask you the closing question that we ask all our guests here on Better Biopharma, and that is, in your opinion, how do you think we can better biopharma? Whether it's in how we design, develop, manufacture biologics, how would you most like to see the industry improve in the short
Yeah, I think the
or
key thing from the standpoint, particularly on the vaccine side of things, is make vaccines that are more user friendly and like ours, which is going to be orally delivered and potentially sent to your home. We think that's going to make a huge difference from the standpoint of public health because the more people that take vaccines, the more herd immunity build, the more healthy our nation will be.
It's a great point, and that prompts a follow-up question in my mind, and this is what we can end on. Um, are you thinking about how to partner with, public health entities in the future to, best case scenario, you get this thing all the way approved, and is the are you in that long-term frame of thinking to how are we gonna partner up to get these in people's homes, and really just
Yeah,
very
I mean, I don't. Again, I'm not sure we're how how to sort of say that we're going to go work with the governments to sort of like to have them market our vaccines. I don't think that works, but I do think that we can think about it in terms of like we think that once you make it very easier on the end user, that'll be ways for commercial entities, things like companies to partner with them to sort of like build get all their employees vaccinated or build things like work with Kaiser Group Health, all these big HMOs that really, really want to sort of improve their public health because it hits the bottom line. The more people don't go to the hospital, the less money they have to spend. So we do think that if goes across the finish line and gets approved, we do think that working with the big players to make sure that more people are immunized is going to be really important.
It's awesome. Well, Dr. Tucker, thank you again so much for joining me today here on Better Biopharma. And thank you out there for tuning in to Better Biopharma, the official podcast of Bioprocess Online. We'll see you next time.